Live from One Cancer Place is a series of conversationally brilliant discussions among physicians, scientists, and some inquiring cancer patients. Our guest expert today is Dr. Aparna Parikh of Mass General in Boston. Dr. Parikh is hosted today by our genial host, Dr. Christopher Heery, the Chief Medical Officer of Arcellx Incorporated. Our series is supported by several industry and organizational partners. Our founding partner is CGen. Supporting partners are Natera and Mirati, and supporting organizations are the Colon Cancer Coalition and the Colorectal Cancer Alliance. Thank you all for all of your support. The floor is yours, Dr. Chris.
Thanks again for setting this up, Erica. Today on Live One Cancer Place, we have another very special guest, Dr. Parikh, who is going to walk us through some of her research interests and some of the things that we are learning in not only GI cancer research, but how those things might apply across other cancers and how we can help educate some of the patients that will be tuning into this and how they can help other patients with these learnings. So I'd like to extend a warm welcome to Dr. Parikh, and maybe hand it over to you if you could just kind of tell us about you, a little background for anybody who's tuning in, and then we can go from there.
Erica, Chris, thanks so much for having me today. It's such a pleasure to be here, and I'm really, really excited to get to have this conversation today. So I'm Aparna Parikh. I'm a gastrointestinal oncologist at the Massachusetts General Hospital Cancer Center. I see patients primarily with colorectal cancer, a little bit of pancreas too, but primarily colorectal cancer. My research interests are a little bit vast. So a couple of different areas where I've spent a lot of time thinking about. So one is just in the more advanced setting and how we try to really bring novel therapies, particular immunotherapy approaches to these GI cancers, which are really, really tough. And except for a couple of GI cancers like upper GI and liver cancer, we just haven't really made much headway, of course, in the microsatellite unstable patients we have, but haven't really made much headway in progress to try to enable these immune approaches to work. But the other area that I spent a lot of time thinking about are these blood-based tests, which is sort of a blanket term of liquid biopsies, which can cover a lot of different approaches, but particularly circulating tumor DNA and cell-free DNA approaches. It's a really, really exciting time for blood-based diagnostics and blood-based tools. We used to use blood-based tools as an adjunct, and we are using it as an adjunct in the stage 4 setting for molecular profiling to really try to figure out, what patients have, what driver mutations, and as new driver mutations emerge, can we pick something up in the blood that we may have not found in the tissue? One of the advantages to the blood in the metastatic setting is that you can much more easily get temporal information as well as spatial information. So you can follow people over time rather than having to do repeat biopsies. And when you follow people over time, you start to understand mechanisms of resistance and new alterations that may come up. So that's where we've sort of focused initially, and I can tell you some more a little bit later about other applications in the metastatic setting. But what's really interesting now is that these blood-based tools are coming into the curative setting too. So we're using it as a tool to try to figure out, which patients are cured after surgery and which patients aren't. And can we use these tests to help guide additional treatment for those who we think may not be cured? And then as these tools become more sophisticated, which they are, and they're rapidly changing every year, can we almost use these as a better tool for personalized medicine in general, right? Can we figure out who may not need additional treatment? Because right now we often treat a lot of people who actually may not need chemo, and we're subjecting them to toxicity and side effects of that. And there was a group that was cured with just their surgical approach alone. And then Increasingly, I'm sure I know you have some other young onset champions coming onto One Cancer Place to discuss with colon cancer. So I'll get off on my soapbox just for a quick minute around the 45 screening guidelines for colorectal cancer. But some of these blood-based tools are increasingly coming into the place in the early detection setting too. So certainly a really, really exciting few years for a blood-based test, not only for early detection, but residual disease. And then, like we said, in the metastatic setting, besides just profiling, some other interesting applications emerging too.
I want to do a quick recap of some of those topics because they tie in well to some of the conversations we've had recently. And it's always great to get another perspective on how these tools might play out, not just for colorectal cancer, but how the research in colorectal cancer might impact somebody with a sarcoma or somebody with pancreatic cancer, so on. One of the things you touched on is this idea of mechanisms of resistance, which I think for an average patient is probably a relatively difficult concept to understand. So I like to try to unwind it a little bit in every one of these conversations, because we as oncologists talk about this and it's just such an obvious thing to us. It's so important. And so I like to use this analogy. Let me know if this works for you, but this analogy of, if you had a million roaches in your house and you were trying to eliminate all of them, right? And just a few you don't kill, because whatever reason, they don't take the poison or somehow they figured out how to not die from that poison. And then they make a bunch of baby roaches. Now you have 1,000 new roaches who are all able to avoid the thing that you used to eliminate. That's the problem, right? And that's essentially the way cancer cells work. What I just described, though, is a concept that most people don't necessarily tie with evolution, but it's actually the core concept of evolution, which is those fit for the purpose, those able to survive in a certain environment are the ones that do and then thrive, right? And so we actually had this conversation with Dr. Kopetz just a few weeks ago around evolution and thinking about cancer evolutionarily. Can you talk a little bit about that concept and actually how those tools that you just described help us identify those things and how they might influence treatment decisions over time?
Smiling. I love that analogy, Chris. I'm going to use that analogy in clinic. I love the roach analogy.
It always grosses people out enough.
I have other analogies, but I've never used that one, and I love that one. So I think what these tools do, and again, stop me if we feel like we're going to kind of over people's heads, and let's just make this a conversation. But what these tools do is exactly as Chris is saying, is when you follow people over time, every cancer has certain mutations that those cancers needed those mutations to drive, right? They can't come from anywhere. And we know it's not as simple as that. It's not just mutations, but kind of as a proof of principle. So you figure out why the cancer came to be, right? And then the cancer continues to be, and then you give a treatment, and that continuation to be is hampered by that treatment. But as Chris was saying, is there's all these other smaller things that we're not picking up necessarily on a routine basis, because they're not the main ones driving it, but all these other kind of persister cells is a scientific word we sometimes use around that. And those persister cells can ultimately start growing and growing and growing. And those are your roaches that ultimately lead to a drug no longer working, right? So the drug isn't kind of capturing everything. You have a billion cells you have to deal with. But you're having these other clones that are growing and growing. And with those clones, if you can't suppress them all, eventually they'll get smarter. And then those will be the ones that will kind of drive the resistance going forward. And what's cool about these blood tests is that with those, you can pick up what those new clones are. So you can pick up and kind of tag those new roaches that are growing, right? So the classic example, and I'm sure you talked about this with Dr. Kopetz, is when you get RAS status for colorectal cancer patients. And if you're a RAS wild type patient, that opens up some therapies that are not yet available to RAS mutant patients. But that doesn't mean everything is like RAS wild type, right? So you have some cells that actually, maybe they're at the get-go that weren't going to be susceptible to that therapy. But then as you kind of give that therapy and drive pressure on those roaches, these other roaches are, growing and growing and growing in number. And those may be, cells that have the resistance alterations because you've sort of taken care of the ones that aren't resistant. And in the blood, you can track that. So you can sort of see what's happening. So the stuff that was there at the beginning in a few months may be like different than what's there because you've taken care of the main stuff, and now the new stuff is coming up, and you can test that in the blood. Does that make sense?
It makes great sense, and I love. Thank you for continuing my roach analogy. Love that. But let me help visualize this a little bit, right? So let's say they're not roaches. Let's say they're beetles, and like the first set of beetles has like really strong pinchers on their mouth, and that allows them to chew through like wood, and that's where they get their food. And then this other set actually doesn't chew through wood. You made your poison to actually be in the wood. And this other set, actually, they find their food some other way. And so what you've done in biology is oftentimes you find a branch point where that selection mechanism of evolution will allow a certain set of features to allow that cell or that roach or whatever to actually improve their functionality.
Yep.
And one of the things that often gets missed in that conversation is like, gosh, that sounds so scary. But one thing we see pretty consistently in cancer is that the push of a given therapy often results in a certain phenotype in the cells that are selected out, right? Not all the time, and it's certainly probably unique amongst certain cancers and certain pressures. But one of the things you're describing is the ability to anticipate what a given pressure might do in a population of patients, right? And then be able to select for a therapy. So now, let's say the ones that don't chew through wood actually like digging in the dirt. Now you'd say, oh, well, then I want a therapy that is ready for them in the dirt, right?
Exactly.
And that's something that I think is a really hard concept if we're talking about it in terms of mutations and gene expression or whatever. But if we think about it phenotypically, right? Like, and when I say phenotype, I mean just like the way things look, the way they behave. It's a little easier for people to understand that, right? It's not so conceptually difficult. And that's the kind of thing we're trying to help the patients that tune into this show to understand. Is like, why do we do these tests? Why are we searching for these things? How does it relate to making the next big decision? So now, taking all of that, like, let's assume that everyone kind of caught that concept. And if not, just rewind. Well, listen again, and then listen again, and listen again. And someday you'll pick it up, because these are not easy concepts. But let's assume that people pick that concept up. Now, from there, what do you do next? As the physician researcher, what are you taking away from that information? How are you building that, not into just an individual's treatment plan, but the research focus that you're going to take across a group of patients? What do you do next from there?
So you start to look at patterns, right? So there's patterns that emerge when you look at this collectively of what thrives and what doesn't thrive. And those patterns sort of drives the next kind of strategy of therapeutic strategies, right? So, one strategy maybe is, there's so many different, in the targeted therapy arena, right? Sometimes there's so many mutations that emerge. Sometimes it's sort of taking the gas pedal off a little bit and giving a break from that therapy. All these sort of, subsets may start to disappear again. And then you can sort of say, okay, we got control again, and let's try again. The other strategies are we just need new things, right? So just trying to target this in the same pathway, and different parts of that node, the different parts of those nodes may not really work. And so let's figure out other ways to intervene. And that's kind of what we're doing in the targeted therapy space right now. So learning, I think we have to understand what's driving it first, and then as we understand what's driving it, patterns emerge. And that's been the beauty of a lot of these liquid tests in different cancers. You're starting to understand where these patterns are emerging. Is it the drug binding at at the cell surface? Is it in the drug kind of in the, at the cell surface? Is it certain binding pockets and different things? And it's fascinating, right? Because we didn't have these tools a few years ago, and then you see these same themes like coming up over and over again, where the cancer cells are getting smarter to try to kind of overcome that pressure of the drugs. And then what we do is we sort of think one step ahead now. And so we're learning from different targeted therapies, like let's be smarter. And now we know where it's likely to emerge, and let's design the next drug to overcome that quote resistance mechanism, as we call it. And that's what actually, when you look at generations of drugs, sort of as a big box, if you look in lung cancer, if you look in bile duct cancers, you'll start to, or these rare fusion events, when you start to think about, not resistance, but when you start to think about next generation drugs, many of these next generation drugs are exactly trying to overcome those existing and known resistance mechanisms that we many of times just picked up in the blood.
I think one thing to highlight here is, as people are watching this, I hope everyone can see what I see, which is just the incredible passion about these concepts. It doesn't come naturally to everybody. There's a lot of just getting beyond the barrier of the language here, right? Understanding that, just like learning any other language, which I've never been successful at other than medical language, once you get to just a conversational point, all of these concepts are they're so similar to other concepts we use in our daily lives, right? We just need to get to understand what is it that allows this thing to occur. One of the things you mentioned already was the concept of using, molecular tests from the blood that can help us to identify these evolutionary events or whatever much, much earlier. Can you talk about why that is important, and then sort of put it in context of maybe the roach analogy or maybe some other, but what advantage do we gain by being able to figure that out earlier in the process? What do patients take away from that?
Yeah, so I think the advantage of understanding it is exactly, I think a few minutes before what I didn't say very clearly, is to try to figure out that next way to overcome it, right? You don't know what you can overcome until you know what you're up against, right? And so once you know what you're up against and how that mutation or resistance thing is happening, then that's what leads you to be able to figure out your strategies. I think I'm trying to, maybe Chris, you can help me here. I'm trying to figure out a layman's term to explain a gatekeeper mutation, right? And I think that's a great example of a place where we know a mutation in that spot can lead to resistance, and the approach. Yeah,
I don't know if there is a layman's term for this. I keep trying to think of an analogy too. I mean, so the concept for the audience that we're talking about is this idea that there is one central feature of that cell that allows it to survive and thrive, right? And whatever that is, it is going to influence the way that cell behaves. But one of the things that we tend to do when we talk about cancer cells is sort of go through this anthropomorphic view of it. These are so smart, right? They're not smart. They're just cells. And cells are just running computer programs, essentially, right? That's in DNA. And when that happens, if there's an error in that code that allows that cell to have a certain set of features, and that works out for that cell to work better, then it will survive and make more of itself. And the weird thing about having ultra fast evolution is that there's unlimited opportunities for that to occur, right? It's just happening over and over and over and over again. And so that's why when we talk about curing patients with cancers, we usually have to come at that cancer from multiple different mechanisms to kill cells, right? Because we have to assume that some proportion of those cells already have some built-in way to overcome one of the mechanisms. So we need 2 or 3 or 4. So you notice, like when we give adjuvant or post-surgical therapy, those are almost always with at least 3 drugs, whether it's breast cancer, whatever, right? So colon cancer is maybe not the best example, although it's close, right? Like maybe someday it will be. I think that's exactly what we're getting at, is that if we knew earlier which patients were going to have their disease come back and why, we would customize the therapy to that mechanism, right? Is that helping at all, or is it driving us away from what you were talking about?
No, it's like super, super helpful. But I mean, just they're confusing, and it's like it's hard to really, it's hard enough for us to get our minds wrapped around it, right? That's why we're in this line of work. If it was easy fix, we wouldn't be here.
Right. Well, great. So those are, those are super powerful concepts. I want to go a little bit down the road of one of the things you touched on first before we get to kind of targeted therapy, because they are a little bit branched in my mind, and that is immune-based therapeutics and what some of the challenges are there. So you touched on that in GI cancer, we haven't seen a tremendous amount of impact with, for instance, checkpoint inhibitors, which have had an enormous impact over the last 10 years across so many cancers. First, could you sort of touch on some of the strongest hypotheses behind the whys there, why that hasn't been the case, and then what some of the challenges are that we need to face? I will mention here that I have an enormous amount of opinions on this. I'm going to reserve mine until I've heard yours, and then we'll discuss them back and forth a little bit. But this was my area of research when I was in academic medicine too. So I would love to hear, like, where do you think the state of the field is? Where have we come in the last 10 years? Because we've known this for about 10 years that colorectal cancer doesn't really benefit much, but where are we? What are the challenges? And what do you think the next big steps are?
Oh, pressure's on. I'm curious to hear your take on it.
Well, mine haven't changed in like 8 years. So
well, unfortunately not.
Sadly.
Colon cancer. But I think the way you want to sort of think about the immune system too is there's so many different components to it, right? So there's the tumor component, right? And the way I sort of like to explain it even to my patients, and you'll hear this in the lingo, right? Cold tumors, hot tumors, and what is cold and hot referring to? So kind of cold tumors are generally referring to the tumors that I like to think of as like the Harry Potter cape a little bit, like they just hide themselves really well, right? So, and that the reason that they may hide themselves really well is because innate to that tumor type, like colon cancer, there's just not a lot of stuff to be recognized as kind of foreign, right? So the kind of trigger signal for the immune system for all of us is the way we fight colds and COVID-19 and all this stuff is, you need that signal to sort of say like, hey, I'm here and I'm not supposed to be here. And like colon cancer doesn't have a lot of those signals inherently. And what's interesting is, especially for some, environmentally exposed cancers like lung cancer, that there's some association with smoking and UV light and different things. Like there may be some environmental exposures that kind of help a cancer be recognized more and more too, but that's not enough. So there's some kind of fundamental things to that cancer. But then also that cancer sort of doesn't sit by itself. It sort of sits in this like micro environment and macro environment. So there's the kind of micro piece and then there's a macro piece. And there's all these different layers of protection. And so even if then you had a cancer and you were able to figure out, okay, there's ways that I can maybe get the immune system to recognize it, that's all well said and done. But if you don't have the right cells to come and recognize it, like those are your T cells. That's what we talk about. If those cells aren't there and near the cancer to do anything about it, it doesn't matter if you can show it here and then those are over here. So you got to figure out not only to kind of unveil your cape and show yourself, but then to get all the right kind of parties that need to be there to actually take care of it as well. And so it's complex, right? It's not just the tumor, but it's also what's happening with kind of the immune cells. And so the way we're sort of thinking about it is it's a multi-pronged approach. So like, it's not going to be enough just to like show yourself to the immune system. In some cases it is, like the microsatellite instability patients, because of the way these microsatellites engage their immune response in such a profound way. But we know in those tumors, there's lots of T cells already there, right? So because of that, those are, those cells, those fighting cells are kind of there. So you need the tumor itself, but then you need these fighting cells to kind of come in and kind of actually do their job and work. And that's what a lot of these checkpoints are, right? Because those checkpoints can kind of prevent those cells from doing their job too. So it's the tumor and the cells, and you need both to kind of work. And in colon cancer, unfortunately, most of them we don't see either.
Yeah, no, you touched on so many of these like topics that they're really, really difficult. That's like the simple version, but I mean, how our immune system recognizes tumors is really difficult, particularly, and this is true across almost all cancers, even though we've had tremendous success in a select number of cancers, the vast majority of patients still don't benefit from immune-based therapies. I think that's probably something that gets lost in the shuffle of this conversation a bit. But as we just talked about the complexity of sort of evolutionary biology in cancer, one of the things I've always loved about using the immune system to attack cancer is that the immune system is also capable of accounting for the evolution of cancer cells, in theory. That's a really, it's maybe the only mechanistic approach that we have that could be able to account for any evolutionary path of cancer. The challenge is, it's not that simple, right? So cancers can downregulate the targets that we think we're going after. They can put up shields. They can do lots and lots of things. So in colorectal cancer specifically, I know that this is a huge challenge, but one of the things I hope that any patient or anyone else tuning into this episode takes away is that the work being done in colon cancer is applicable to the other roughly 70, 80% of patients in other cancers who do not benefit from checkpoint inhibition, right? Like that, a lot of these concepts are very, very similar. They might have nuances between the different diseases and even within patients, but understanding those mechanisms is what's going to allow us to benefit. Instead of 20% go to 30 to 40 to 50 to 60% of patients, or 100% someday, hopefully. But I did want to dive into one other thing that you kind of alluded to, which is coming at it with multiple mechanisms. Right. So over the last 10 years-ish, one of the other concepts has been to combine things like small molecules with immune-based therapies. And one of the topics that comes up a lot is, in particular in colorectal cancer, is that there are overexpression of certain genes that tend to prevent T cell function. So particularly we talk about TGF-beta a lot in colorectal cancer, but there are a couple others. Could you share just your thoughts on some of those pathways that might be super relevant in colorectal cancer and how they might apply in other cancers and how people are thinking about them now?
Yeah, no, that's a great question. But I'm also, I think one thing that you said that really struck me, which I think we don't think about enough, is like, the immune system's role in even preventing a cancer development too. So there's some immunologic controls that's happening, even at the outset. And it's not like our adjuvant chemotherapy is working for everyone, but, there's a cancer in there. And so those patients that are cured, is that purely just function of removal, or is there some immunologic kind of surveillance that's happening that's kind of taking care of it? So I think it's a really great point to sort of think about it as a kind of continuum of immunologic control or lack thereof that perhaps ultimately, even led to development. So you touched on a one that's really interesting in colon cancer, probably about a fourth of colon cancer, really important in pancreatic cancer too, that in some others, cholangiocarcinoma and things too, the TGF-beta pathway. And what we know with sort of TGF-beta is that the TGF-beta kind of in that microenvironment sends off these like suppressing signals. And those suppressing signals can actually impact the ability of T cells and things to actually get in. So TGF-beta is one, and there's kind of many TGF-beta approaches being developed. And some of that is also all the TGF-beta can actually contribute to that what's called the stroma. So stroma is sort of the thick environment, like to think about it, that the cancer sort of sit in. And if you have this like really thick environment, how are you going to get stuff in? And that stuff includes treatments, but also immune cells. So if you can kind of take care of that pathway, maybe you can allow, anything that you're trying to use to treat that cancer to work a little bit better. We actually have a very exciting trial that we're waiting for the readout for the next couple months in pancreatic cancer, looking at a blood pressure medicine that targets sort of TGF-beta to see if that helps. And that came from some early work in our group. But another one is, I'm sure Dr. Kopet's talked about this too, because, his group is really kind of leading the kind of MAP kinase work along with others in our group too, like Ryan Corcoran. But combined, like we think that signaling through MAP kinase, that MAP kinase pathway as well, may be sort of another immune suppressive approach that's leading towards, again, sort of T cells and others to not really signal as well. And so if you can suppress that kind of pathway, then you see these other gene signatures come up that are looking like pro-inflammatory. So we're starting to recognize that's another pathway where signaling by that may actually hinder immunologic mechanisms. And then if you can kind of really target that pathway, then perhaps you can engage these immunologic mechanisms.
Yeah, I think one of the kind of amazing things that has come up as our diagnostic tools and our, just the assays that we have available to us today that have come up, and our ability to do these big drug screening programs, is that as we start to understand the complexity of the interactions between the cells that are within a tumor, the actual tumor cells, the things that we, if we cultured it out and grew it in a lab, we would say the tumor cells and the immune cells, they're not actually distinct from what we think of as cancer, right? The cancer that we see on a CT scan is actually a combination of a lot of different cell types. The cancer cells in some cases may end up being only 15 to 20% of the mass that is causing the problem. And one of the challenges I think we face, pancreatic's a good example, is when the stroma makes up a large proportion of the tumor, how do you get rid of something that is normal cells that is intermeshed within the cancer cells? Obviously, you have to get rid of the cancer cells, but so one of the things we're kind of talking about, and this is maybe a side note, a different concept, is understanding the systems of cells and how they interact with each other. And one of the things that's come out of that is these big drug screens don't tell us if we look at one cell type. We have to actually combine lots of cell types to find these things.
And I think beyond that too, it's the larger environment. Like the liver is probably the liver is different than the lungs, different than the belly cavity. And so you take that organ too, and that cancer may be very different the way it sort of acts in that organ versus other places. And so I think like more and more as we're getting smarter about this, I've said this, many times before, it's sort of cutting the pie up in many different ways. And it's like definitely not this one size fits all approach. And this one size fits all approach, when we used to think about it that way, it sort of led us to failure after failure. But I think we're getting smarter of understanding that it's like there's so much contextual stuff that matters. And just understanding even the tumor microenvironment in one organ may be very different than in another organ too. And so again, another. We keep digging, hopefully without making it too complicated for the audience, but we're digging into like these concepts that I hope this makes sense about why it is so hard to cure cancer, right? So another way to think of this is, if you take me and then my brother and then my other, and we end up moving to different parts of the country, right? We did that because there were certain things about that place that we liked better. It fit us better. Similarly with cancer cells, if one of those cancer cells ends up in the liver and then grows more cancer cells versus a different one that ends up in the lungs and grows more cancer cells, there are probably some things different about the cells that planted themselves there
that allowed them to thrive in those different environments, because the liver has a very different set of signaling pathways and, the way the cells interact with each other than in the lungs, right? So if I planned communities and I moved to Florida, I'm probably a pretty different person than someone who wants to live in New York City, right? And in doing that, I have to think about different food supply and different, and the same thing's true for cancer cells. So as we think about treating patients with metastatic cancer, one of the things we've already alluded to but didn't say specifically is it can be really tough to eliminate every location of that metastatic cancer if there are multiple sites around the body, because they actually might respond differently to the therapies we might use. And we see that, right? I mean, how hard has that been, especially in colorectal cancer, thinking about liver-based mets most frequently? Is that a huge challenge?
Yeah, it's a huge challenge. But I think what's also cool about it in some ways is sometimes you have just one problem spot, right? And so we're seeing this approach where if you have one or two problem spots that, a lot of other stuff is responding, but this problem spot is not responding because it found its way there and it's thriving in that environment, we can think about approaches to kind of take care of that problem spot. And, it's purely research and scientific, but what's cool for us, people that meld the science and the clinical care, is that we can sometimes see in the blood those problem spots emerge, and then you take care of that one slot and you're like, oh, I think we took care of that. And so we very much try, I think, to not to treat every person and every disease as an individual, but not kind of lump them all into one size fits all, even for that treatment, because you may have very individual approaches based on that that particular person's kind of pattern. But
do you think we'll? Sorry, let me let you finish that thought.
Oh, I was saying back to the liver stuff. I was sort of kind of for some talks recently, I was sort of just pulling through some of these different novel oncology approaches with different small molecules and things like you were saying. And there's been some published data around this too. And I was sort of looking at this liver thing. And I think a couple of people have commented on this in the past and in the academic world, is that some of these, if you look at the responses to some of these immune drugs, you're sort of seeing this little signal that maybe if you control the liver, the responses you may get responses outside, right? Or the response, these small signals, 1 in 10 people are responding or 2 in 10 people are responding. There seems to be a little bit of an interesting kind of signal emerging that maybe the responses are happening in people outside the liver. And so, do we think about that differently? So treat the liver one way and then treat the other another way. And my biggest, I think, disclaimer I always like to tell people, and Dr. Kopetz, who has a lab and is a true lab scientist, I feel like a lab imposter all the time, never set foot in a lab. But I sort of see clinic as a lab too, because as you're doing these things, you're seeing these observations, and you have these tools to help you observe, which is really cool. But the even the clinical observations, if you start paying attention, there's themes, and then you try to pull those themes back into all the smarter people that are doing the basic understanding of that. But I think it's cool.
I love that. I think the thumbnail for this episode should be the clinic is my lab. No, I'll say I'm obviously very biased because my work in academics was very, very similar to yours. I didn't have my own lab. I didn't like the lab, but the people like you who can tie together these concepts and implement them in the clinic are incredibly valuable. It's it's a really hard skill set. And so I thank you. I'm sure the patients do too for what you're doing to connect those dots. I wanted to ask you something though, which is, this is purely hypothetical and it's really just an opinion. So we can cut it out if you don't, but as we see therapies improving and understanding molecular targeting better, do you think there is a world where targeted surgical interventions or cryoablation or radiofrequency ablation becomes more relevant in metastatic disease across other tumor types? Maybe just whatever you think.
Yeah, and I guess I'm biased in the sense that we have amazing people that are thinking about local approaches, and we have a strong radiation oncology department, really strong interventional radiology department for lungs, for liver. So like I'm a believer, and I think there are prospective trials going on to look at kind of local therapies in different settings. I think kind of going back, I think what's always humbling about it, and I think what's a really hard conversation to have with people is sometimes just the biology of the cancer sort of tells us when that's the right thing to do and not the right thing to do. And that's really, really hard, right? Because people hate sitting on things, right? But you go in for something that's like very out of the box or more aggressive or more local. And you do that if you think it's the right time, and it's not going to hurt them and set them back, because and I think that's and even with patients, it's one of the hardest sort of concepts to sort of get your mind around, because yes, there's absolutely a role, but there's maybe not be a role for everyone. Because if you keep buzzing things, for many it can work, right? But for others, you take care of one, and then next one comes up, next one comes up, next one comes up, and then before you know, you kind of have this kind of army of stuff that you can't control. And then so it's absolutely, but again, it's sort of individual. It's an individual decision, and you can't. It's again not going to be a one size fits all, because
I think that's the challenge. Is it's very hard to think of sort of like a standard protocol by which these types of decisions get made, right? It is very individual. I'll give you an example of why I asked the question, and then maybe it'll make more sense for the audience. But some of the researchers that were at NCI at the same time I was still working there wanted to look at mechanisms of resistance for erlotinib in EGFR mutated lung cancers. And so what they would do is, once patients reached their maximum depth of response, they would take that patient to a surgical resection of any mass that was left over. And their goal was to identify what other targets they could create new therapies against, right? That's a very interesting scientific question, and it resulted in great work that has led to new drugs. But along the way, what happened was, all the patients that they cut that tumor out, some of them never had their tumors come back. And they were like, wait a minute, maybe we just took out the last resistant cells, so they couldn't make more resistant cells that later had more problems that were harder to solve, right? And what they were just was staring them in the face, which seems kind of obvious in retrospect, is if you take out the worst of the cells before they can make more bad cells, maybe you cure some people. And it's too early to say that those patients are cured. It's still only been 5 years since then, but still, it's pretty incredible. And I just wonder if we see that world coming, right? Where we have to rethink our paradigms about when and how. We've done this in oligometastatic colorectal cancer for years, but is that world going to start to rise up? What do you think about it that way?
Totally. I mean, we're even thinking about this now with select pancreatic patients, right? When biology tells us this is the way to go. And, in colon cancer, I think the liver met example is a perfect example, right? Just by the time you cure people, right? You cure people with multifocal spots of liver disease, because you just, like you said, you're just taking out it's sort of that big question of, do metastases metastasize? We're realizing probably, right? So if you sort of take out those metastases, not in all cases, but in many, that you can kind of take care of it. So I think the oligomet approach, even in different organs, is going to move beyond colon cancer. And we're seeing that there's lots of that data being generated.
Yeah, I guess we should acknowledge here, for anyone listening to this conversation who might be wondering, hey, I only have one spot left. Why don't we just cut it out? Or why don't we do this? There are a lot of surgical safety considerations on that too, right? So what people always have to balance is, what is the likelihood that this is going to clinically meaningfully improve your overall lifespan versus what is the risk I might kill you by doing the surgery, right? That's a serious consideration. And that's part of the reason why this has been happening in colorectal cancer for a long time, is that it's easy to cut things out of the liver relative to some other organs, not always. I don't want to make that like seem like everything's surgery is not easy ever. But relative to like cutting something out of the lung, sometimes that's just a safer surgery to do, right? So bear in mind, when you think about this question as a patient, don't start from the assumption that anything anywhere in the body can be addressed. Like that's not true.
Yeah, so important. We're getting better at other approaches besides surgery too, with pretty good control rates too. So people are being like that field is moving too about non-surgical approaches for control too.
There are a couple of things I want to just like check the box on that we'll probably have to edit back together. So one is, can we come up with an analogy for hot and cold tumors that we could maybe plug back in when you mentioned it earlier to help people understand that concept? So maybe tying in weeds in a garden or insects or something that is like, that would be helpful as we think about what is coming after them, right? Like, let's think about it like, an animal with a natural predator. Okay, so in this case, the immune system is the natural predator. The animal is the cancer cell, right? So let's think about it like rabbits, because rabbits, we all know, reproduce very quickly. Yeah. And then let's say like foxes. Okay, so the immune system coming after those, the fox trying to get all the rabbits. Well, let's say that you have a rabbit that happens to live in an environment where it's just really good at blending in, and the fox's vision just isn't good enough to see it, right? Mm-hmm. Like you've seen the rabbits that live in the Arctic, and they're white, so nobody can see them, right? You have to think about the way that a cancer cell or a mass of cancer cells works. One, as being able to pretend like it's normal, that's one thing. So cold tumors generally share one of two things. Either one, they're able to pretend like they're normal well enough that the immune system doesn't come after them, or two, they're able to fend off those things. So in this case, now the rabbits have built a fence that keep the foxes out.
No, that's right? Yeah. I like that analogy. So it's like the camouflage, right? The camouflage of that Arctic white rabbit versus okay, camouflage isn't going to be enough. So like, let's put up that fence. I like that.
Yeah, we're just making it up on the fly here. But like, I mean, I think one of the things for patients listening in is to think about why our immune system doesn't go after a cancer generally comes down to the mechanism by which that cancer became metastatic in the first place, oftentimes. And so one of the huge advantages we have in melanoma and lung cancer and a couple others is that it turns out that in those cancers, the reason the immune system doesn't eliminate them in some of those patients is because of this thing, PD-1 and PD-L1, right? And it's just that thing. And when we break that cycle, the immune cells kill them all. Well, that's fantastic. But what about the other 80% of them, right? And that's where we are. So hot and cold tumors, when we talk about it, we're talking about hot means the immune system recognizes it, it's there, it's ready to go. It just needs that last trigger to get over the edge. And cold tumors mean like there's really just nothing going on. Like either it's not being recognized or it's keeping the cells out or something like that. Does that help, Erica? Somebody from a patient perspective, let me know if we're on the right track here. So that was one. The other thing, the other question that came up that I wanted to just pose to you, and again, this may be a bit too specific for the audience or whatever, so we'll see how this conversation goes. But this is from a patient who unfortunately couldn't join the conversation today, and obviously is asking this question from their own perspective, but we want to generalize it as much as we can. So you are personally working on some of these studies for BRAF. You're also working on sort of the CMS subgroup understanding and how you're involved in the investigations of drugs that are specifically targeting TGF-beta. You're involved in those for the MAP kinase pathway and BRAF. Do you see first like an overlap? Do you see anything exciting happen in either of those or anything that actually addresses both at the same time? Because it seems as though there are patients that fall into both categories concurrently. And I'm wondering if you see sort of an overlapping mechanism that might be able to address both concerns.
So I think we're seeing some really, really exciting data of combining these different approaches with immune approaches, right? So is that what you're kind of getting at?
You can take it wherever you want. I mean, this is obviously a big question, but I'd just love to hear your thinking about it.
Yeah, so I think, when we sort of take it back to the issue of when you start to get lots and lots of resistance mechanisms for kind of targeted drugs, sometimes you just have many, right? So you have so many different ways, so you just need an entirely sort of new mechanism. And when you have a kind of fundamental pathway that has an ability to modulate the immune system, that's where we're sort of heading. And what's tricky with some of these targeted therapies is that we know resistance is going to happen. We know it's going to, and we can anticipate that. And for some of these targeted therapies, what we see is that the time on that treatment is quite limited, right? So yes, you're getting a great response for a period of time, which is awesome. And we're seeing some great responses and some really exciting data with some of these new targets. But then pretty quickly after, those, all those roaches are kind of coming back with a vengeance. And so we're realizing that probably targeted in and of itself is not going to be enough. And so that's why we're looking at, which pathways in particularly can also have an immune mechanism. And then can you capitalize on the targeted therapy to kind of help kick in that, cold tumor to make it hot, and invoke then the immunotherapy to try to kind of mop up what the targeted in and of itself can't do. So that's how I like to sort of conceptually think about it. And just the problem with, I think targeted therapy is I work in this space too. It's really exciting. And the fact that you can kind of predict and make next generation kind of targets available. But what has always just really troubled me is that resistance question, right? So at some point in time, we know that's going to happen. And you can, for many of these, FGFR and cholangiocarcinoma, we've gotten first, second, third generation FGFR inhibitors that keep getting smarter and smarter. But we're still not curing it, right? We're still not entirely sort of taking away, those ones that have found themselves to thrive despite that. And those are the driver ones. And so we have to think about these combination approaches, but have to think about it in a way that's mechanistically makes sense. You can't just we know that people try to combine PD-1 with everything. And it's not working for the far majority still, but there are these subsets where I think when we're smart, as we become smarter and smarter and understanding, I think that this is what I think people in the race, even for patients, I think what's important is when you're kind of signing up for these trials and things too, that's what some of these biopsies are trying to do. When people aren't trying to biopsy, they're not biopsying for the sake of biopsying, right? I think really trying to understand for both the people that respond and when you don't respond, what's happening with this complex interplay of tumor, microenvironment, organ, that's leading it to kind of become resistant, and then what can we do to make that better? So that's a little bit of so my answer is yes, combination approaches are clearly coming, and we're seeing that with BRAF and IO, and kind of 2 trials out there right now where we're seeing longer durations of responses, which is exciting. Not yet kind of curing in that space, but we're seeing that. And then TGF-beta is one component, and we're seeing TGF-beta IO combinations and drugs that are combining the TGF-beta plus the PD-1 in sort of one drug. So absolutely.
Yeah, I just would love to add on one little comment there to something you said, which is the biopsies that are asked for in clinical trials are, I don't want to overstate this, but they may be the most important part of a contribution that any given patient can make to the scientific field, right? There is nothing more valuable in what we do in research to understand all of the stuff we've been talking about today than getting tissue. It is so critical because, as we've talked about today, the mechanism of what drives killing a cancer cell may be true for 30 or 80% of the cells in that biopsy mass, but it's the ones that are still left behind that we need to understand better. And that's the difference between being able to make informed decisions and uninformed decisions, right? And I hope people see from outside of the cancer research world is that the pace of innovation is exponentially increasing over the last 30 years or so. And the biggest limit to that pace of innovation is getting the right samples to be able to make the right inferences and ask and answer the right questions, right? And so I'm not suggesting that every patient should rush out and go find a way to get a biopsy done. Get a biopsy if it's asked and if it can be done safely in the context of a clinical study that is asking real questions and has a real hypothesis behind it. Please, I mean, that's like, I will say that. You don't have to say that, but I will say, if you can manage it, please say yes to those. It's huge. It is hugely important to the answers to these questions.
And I think I also want, I think we're thinking about this approach ahead of time, right? So even some of the smaller 20, 30 patient investigator studies that we do, like the way our team sort of thinks about it is like, what are the questions we're going to get from this? And like, let's kind of have all our ducks in a row, like before we dive into this. So like, even if we're helping a small number of patients, like those deep learnings that we can generate from some thoughtful planning about kind of mechanism and things kind of leads itself to the next trial. So it feels slow still for patients, right? And it feels slow for oncologists because we're not, especially in GI, we're like feeling left out and we're not quite there yet. But, I think people like, us are trying to chip away at it slowly.
One other question that Erica just put in the chat, which again, we might be able to kind of splice back into the earlier part of the conversation, is like, could you help define mechanism of action and mechanism of resistance for people just like at high level? What do we mean by that?
Yeah, so the mechanism of action of a drug is the actual way that we think a drug is working, right? So what is the target and how is that drug engaging the target? And there's so many different classes of drugs. Chemotherapy has a different mechanism of an approach than immune approaches, than some of these other approaches that people are hearing about, like CAR T cells and things too. So it's really thinking about the way that you are engaging the cancer, I guess not cancer necessarily, but I think both the target. But it's, I think some of these, the other class of drugs that you might hear about are called like antibody drug conjugates, ADCs, right? So that may not necessarily, it uses other components and other parts of the cancer, so certain expression of certain proteins, for example, to bring in something that can then elicit a response. So when we're talking about mechanisms of actions, we're basically talking about the tools that we have and how those tools actually engage the cancer. And it's not always just the cancer cell, it's on the cell surface. It's the proteins around the cancer. It's, so lots of different, some things internalize it into the cells. Some are just at the receptor because they have certain pathways that are being kind of signaling via these receptors at the cell surface. So that's what a mechanism of action is. And then the mechanism of resistance, again, it's a general term for all the different ways that these sort of start to evade the different approach. And so in the immune, so we talked a lot about targeted therapy, mutations, and mechanisms of resistance, but there's also mechanisms of resistance for immunotherapy such that, it requires a certain cell to present that foreign entity, the cancer or an antigen, to the immune system. And those cells that actually present that antigen can become mutated in a way that they can't present it as well, too. And so that is the mechanism of resistance for that approach. So it's cancer specific, but it's also it's drug specific, depending on the way that the drug is trying to kind of take care of the cancer. Does that make sense?
It does to me. Unfortunately, I
I know.
These are concepts that are so natural, I guess, in our world that I want to make sure from the patient perspective that is relatively clear. Erica or anybody else, what do you think? I mean, do we need more on that?
I just want to say thank you. One of the things that I have discovered in my role in my life in trying to discover as much as I can about cancer and the experience is that you all speak a different language, and learning the jargon and what it means is really, really important. That's where, Dr. Chris, you do such a beautiful job at creating these metaphors. I mean, it really makes sense. And I have heard the term mechanism of action for at least the last 4 or 5 years and not been able to distinguish between mechanism of action and mechanism of resistance, which is why I asked that question. So thank you, both of you, for allowing us to ask these very simple questions just to kind of clear up what we're learning. So I really want to emphasize that. Chris, you do a beautiful job with those metaphors.
I know. I know.
Don't stop. Please don't stop.
Well, I don't know if my kids would agree, but well, I'll take it. Thank you. So one thing I would just say is that, like, when you hear words like this, first of all, Google is amazing. Like, just Google things you never heard of them. Like, go to Wikipedia. You'd be surprised. People make fun of Wikipedia a lot. Like, Wikipedia is a fantastic resource just to get, like, the first 3 sentences of every Wikipedia entry are very useful just to be like, what is this, right? Mechanism, really, you don't have to think about it any more complicated. Like, the mechanism by which something happens, right? Like, if I need to know the mechanism by which I get to work, it's like in a car. This is the mechanism, right? Like, how? And so, like, when we're talking about these things, break it down to what are the words we're saying. The mechanism by which this drug works, the mechanism by which the cells get away from the way the drug works is the mechanism of resistance, right? And there's a lot more complexity to that, but honestly, you don't need to know it right now. It's not going to help you that much. Because the stuff that we spend time on and we talk to our lab friends about, and like how they design the chemicals to hit this one spot, like, if you get to that level of depth, you might want to come work in this industry or in academic research, right? Like, beyond that, like, when you get to that point, you're now asking for grants. so don't worry about it that level, right? Yeah. But just know that these bigger concepts are things that we need to be able to understand. So similar to, like, if I need to know the mechanism by which I'm going to get outside versus the mechanism by which I'm going to get to work, like, those are 2 different mechanisms, but they're, like, easy to observe. I can't observe that in cancer without taking a piece of the tumor or looking at some really advanced test in the blood, right? And that's the stuff we're getting at. I want to say thank you to Dr. Parikh, because this was, for me, one of the easiest hours I've spent in a long time, because she is incredible fount of knowledge here, like, and really, really great at putting this into words that people can understand. I think we can honestly say that, like, you are really clear about how you explain this to patients. You must do this all day, every day, which I'm sure you do. And it's really, like, really, really great to have somebody just explain this stuff the way you did. So thank you for putting it all into context. I hope someday we can have you come back, because I think we could do this for another hour easily. I do want to ask if there are any burning questions that anybody wants to ask before we close it up, because I know you probably have work to get back to on a Friday afternoon. But any other things?
This is Mimi, and I just want to say thank you so much, because I wish that my doctor would explain mutations and cancer the way you all have done. So thank you so much for the explanation. It was very easy to understand.
Thank you so much. No, and I sort of, like, think about it from the lens of, I know Erica knows this because she saw it on social media, but I was, like, with my daughter's class this morning, 2nd graders, and trying to explain, like, what oncology is and what cancer is, and the questions these 2nd graders had, it's like, oh my goodness. But as you start to talk, you're like, this is how we should be thinking about these things, because we assume a level of understanding that, it takes all of us like so long to even get to, and like we just take for granted our language, as Chris said. And so I'm so grateful to be here with you all. And Chris, I was anxious. I'm like, what are we going to talk about? And like you told me not to worry about it, but you made it so easy and fun. And Erica, you assured me that was going to be the case. So thank you.
We're so lucky to have him doing this with us, because he's an interpreter between two different worlds, really. And Chris, you really are amazing at doing this. So thank you. And thank you, Dr. Parikh. This was a beautiful time with you this morning. And speaking for all of us, I know we're thrilled and delighted, and we would love to have you come back and do this with us again.
Happy to. Thank you guys so much. Good seeing you.
Yeah, thank you
so much to everyone.
I know I felt my questions were pretty simplistic, but I feel that, honestly, Chris, in the 20 years that I've been around this cancer business, I've really come to understand that I do need to ask the simple questions about what things mean.
I should tell you, those are not silly questions, Erica. When I started in medical school, like I remember like the first week, people were talking about diabetes, and I just was like, "What is diabetes? Like, what is diabetes?" Right?
Yeah.
Like, because I mean, like, people just assume, oh, you get to medical school, like, you know this stuff. I'm like, they don't really teach you that in,
Grade school.
Well, organic chemistry. They don't teach you what diabetes is. So all the stuff you do to get into medical school, they don't really tell you anything about what medicine is, or what are all these things that people refer to. And that's a silly example. Like, that is so simple, because anyone can now, you can just Google what's diabetes and it'll tell you. But like, I'm trying to remember, was Google a thing? Maybe. Anyway, the point is, I've done the same thing throughout all of my training. So like when I joined an immunology lab, and I was not an immunology expert, there are all these PhDs that are giving presentations. I just go, what's a macrophage? What does it do? And like, you only really have to do that once. And then all of a sudden you're like, okay, maybe I should look that up and read it like 10 more times so I don't feel so silly. But when you say it out loud, it really does make it more real that you don't know what it is. I think all of us should do it more. Just don't be embarrassed about the things we don't know. There's tons of.
I want you to know that thanks to your guidance, I am looking up Wikipedia and Googling a whole lot more, because I really want to know this. I want to know the point. So often I think we find we engage in different behaviors and we do it without knowing what the point is. And I think specifically about engaging in a treatment for cancer. Tell me, Doc, what is the point of this treatment? If it's meets with my thinking, which is that I want to live, okay, but I might want to know more about how I'm going to live and what it all means with regard to me specifically. And so this is the subtext of why I'm trying to get simple questions answered. I want to know what the point is.
Yep, that's so great.
That is amazing. Like, if there's one thing that One Cancer Place could convey to more patients, it would be that concept.
Yep.
What's the point?
What's the point, and how does it impact me and what I want, right? That is not a question not enough patients ask or have thought deeply about what it is that they want. They're just like, well, my cousin told me I should take this drug because of, and you're like, well, why? What do you want to have happen? And they're like, I don't know.
Yeah. Well, I also want to know how it works, thus the mechanisms of action and resistance and what can happen. And so this is why I'm going to keep asking the questions until I fully understand and can turn around and explain it possibly to somebody else. That's the best way to learn.
Yep. See one, do one, see one, do one, teach one, right?
Yeah, exactly. Exactly. I think that's where I've gotten. I thought I could get away without learning it for a long time, but then finally I got to this what's the point idea. And I'm glad you guys like that, because I do think that needs to be preached from the mountaintops. I don't think a lot of people think about it.
And I think I think it's empowering too, right? You just have to people have to understand that knowing this and having some of that jargon And having that toolbox at least talk about these things can actually feel very empowering. I mean, I think even for me, who's now in this translational world, like I think, I had a whole separate life of global stuff and capacities and health system strengthening and clinical care and low income settings. And then because I was like scared to have that, like, I was like, I'm not a lab person. Like, I don't understand this. And like looking at a cell paper, like was just the most intimidating thing ever. And then like, but I really liked oncology and I like oncology patients. And then you just realize like, you have to ask and you have to learn. And like, as you do, you'll be able to like help your patients better. You'll be able to help the field better. And so it's interesting because I think I also find myself drawn to the people who can explain things well for my lab collaborators, because I can sit and like, hold on a second, what are you actually saying? And I remember a couple of distinct conversations where I'm like, okay, hold on, like with cell-free DNA stuff, like what is actually like, we use this word ddPCR as a technology, and like what it like, 6 years ago, I didn't actually, I'm like, explain to me what is actually ddPCR, like what, I don't understand it. And that's like, you have to start understanding and feeling empowered to ask the questions, even we do. And like, if you don't, then none of us are going to move forward until we're asking the right questions and hard questions and pushing people. Because when you push people too, then they're like, oh, like, I don't understand that. And then I should understand it more.
One of the things that I want to show here in what I call these brilliantly conversational productions here is that I'm presenting you all to the world as I know you, which is human. You're totally human with a passion for helping us. And you're not separated from us by all your knowledge. I mean, you know a whole lot more than we do, and you are caregivers for us and everything, but you're also teachers for us, and you're totally human. And it's that humanity that I like to bring to the table here and demonstrate to people. And I think is the very heart of what we're doing here with this Live from One Cancer Place idea. Couple of people have said, gee, these conversations are really long. Well, yeah, they are. And where else are you going to get a whole hour with your physician and your cancer oncologist? Come on, and get a chance to learn like this. So I'm so grateful that you all are willing to do this with us and kind of experiment in new territory, really, in that bring you to our table as people, and that we know and love, and we do. So thanks.
Thanks so much, Erica.
Yeah, it's fun for me. So I guess I would talk to a wall about this stuff. So
Chris, you do a beautiful job. It's wonderful.
But yeah, I'd love to do that, to have a get together and just be able to have a less formal, less whatever this is situation. But obviously, ASCO is always a bit crazy, so I'll try to be flexible to find a way to make it work.
One thing I wanted to add too, on top of your statement, Erica, about what's the point, I think even before that, so many patients don't even have a freaking clue what questions to ask, and or they're so intimidated because the doctors are the experts, and how dare you question what they suggest. I think some education around how to ask, when to ask, why to ask, and giving people permission, because most people aren't like all of us here on the call, where we have no qualms about asking or probing or what have you. But my experience with my brother recently with his liver cancer, they didn't ask. They just went along with whatever anybody told them.
Like, I always just tell my patients at the beginning when I meet them too, like, if you want to go talk to, like, if decisions are overwhelming and they're hard, right? And like, you come and see us and you entrust us with your health and help guide you along the way. But like, I also empower people to like, go talk to someone else, right? Like, we have a couple of great amazing oncology institutions right here in town. And so like, you go see someone else, you hear their perspective, the way they explain things and the way to talk about it. And like it may click for you in a different way. And I think that's another thing that like, I always feel like patients feel like sheepish about, even in terms of like asking. And then like, I'm like, if anyone ever tells you not to get a second opinion, like that's the wrong doctor, right? Like go, like, as long as it's not, for some people, too many decisions and too many people can be overwhelming. But I also like to empower people that's like information is knowledge, and it's your health. And if information, even outside of your oncologist, you need more, you need to explain in a different way, like go for it. And then I'm here to bounce those ideas off together and like kind of come up with the plan that makes us all feel good. So I think taking it beyond just knowledge, like just even asking, I think it's also okay to encourage people to like seek out the people that can give them that knowledge.
I'm looking forward to the day, Dr. Parikh, when every oncologist, every cancer treating physician includes in their repertoire, seek another opinion.
Yes.
You know, really, because that gives the patients permission. They shouldn't have to ask or be concerned about whether or not that's considered, a slap in the face of the oncologist. And I think that'll make a huge difference. All the docs that I know do that. And it sounds like you do as well as part of your opening statement, practically.
Yeah.
And it should be, it really, really should be, because we need to learn. That's the only way we're going to be able to find out what the point is.
Exactly.
You know? So.
Might be one of our mantras. Any doctor who doesn't want you to get a second opinion, don't trust them.
Right?
Run. No, run.
Yeah,
Run.
We've said that a couple times on this, on various episodes of this thing. So I think that's such a great point. I really worry when somebody doesn't want a patient to get a second opinion. It very much concerns me. And also, if you ask your physician to explain something in normal language and they don't even try, you're going to have a hard time communicating over time. It's going to be really, really tough. Now, if they try and fail and they say, let me think about it some more, I'll try again next time, then at least they're on the journey with you, right?
Right.
If someone doesn't even try, it's going to be tough. It's going to be a tough time. I actually don't think the bar is that high for what makes a great physician in general, great oncologist in specific, is like wanting to be there for what the patient needs is like by far the number one thing. And that's like often lost because people feel trapped by needing to do billing and get through their day and all that other stuff. But, I think that's one of the messages we want to get across is like, because people ask us, how do you pick the best oncologist? And you don't need the worldwide expert in BRAF. You need someone who will ask the worldwide expert in BRAF the right question.
Right.
And be willing to think and listen and learn when they come across a new challenge. Let's say thanks and let everyone go. And looking forward to see how this comes out all edited down. But thanks again, Erica.
Bye, everyone.
Thank you
Thank you so much, everyone.
Bye.
All right. Bye-bye.
Thank you. Bye. Thank you.
Thank you.
Bye-bye.
So, wasn't that great? We have such great conversations in our live, brilliantly conversational discussions, I think. So before we all depart for our busy day today, thanks once again to all of our generous supporters. CGen, Natera, Mirati, the Colon Cancer Coalition, and the Colorectal Cancer Alliance. Our patients are so grateful to you for all that you do for us. Please, before I lose you or we lose you, a reminder. When you go to see this on our YouTube channel, please like and also subscribe. We need your subscription so that we can get our names on the channel. So we're grateful to you for doing so. Thank you. Don't forget,